versions, just let me know. Okay, so we have a number of instructors in this course, not just me. Just to give you a little bit of background about me, I've been part of the entomology and plant pathology department for 5 years. I did my PhD at Clemson University, and I was a computer scientist by training, and then kind of picked up biology as a graduate student. But I've seen lots and lots of people go the opposite direction, where they started out as biologists and they just fell in love with computer science and computation. And so it's definitely a two-way street. You can come from either background and really be successful. So if you have any questions, please let me know before, after, during the class. Part of my appointment here at UT, 50% of my time is to be collaborative and to help other labs get their computational analysis done. So I always try to be available for people who want to come talk to me. Matt, who's in my lab, is employed to help people run computational jobs. So if you need help, you can always come to us. I will say, if I don't respond to an email in a week, just send another email. I try to be really good, but sometimes I get overwhelmed. So please don't hold that against me. And if I don't respond the first time, email me again. So that's me in a nutshell. I work mostly on plant genomics. I work a lot on different tree species. We run a genome database. So that's our expertise, mostly DNA-seq, RNA-seq. I will upfront admit I know almost nothing about metabolomics and proteomics. So if that's your problem, maybe don't come to me. But I can give you some names for people to talk to. Next up, Abdullah is in the back. If he can, he's raising his hands. He is our resident Linux expert. He writes a lot of software code. He also runs all of our websites and our mobile apps. So he's a really great go-to for anything as far as Linux. Matt Huff in the back. He is bioinformatics, really an RNA-seq expert, did his master's at the Medical University of South Carolina. He'll be teaching a few sections. Fong Liu is also in the back. She's one of my graduate students. She works on microbiome, and she does a lot of amplicon sequencing. So if you're 16S, ITS, metagenomics, talk to Fong. And Shaylee Yu, also in the back. They all sat in the back. She's another PhD student from the GST program. She's doing a lot with RNA-seq of fruit trees. She's working on peaches and apricots. We'll actually be using some of her data for the course. Nick isn't here yet, but he'll be coming to from the ACF, our Advanced Computing System, to talk to us. Ken, did you have a question? Sure, yeah, you guys can call me Meg. So that's a really good question. I would say that there are a lot of good ideas and maybe not a lot of firm answers to that question right now, because right now the bioinformatics faculty are embedded in all different departments. Ag campus, as well as over in the College of Arts and Sciences, even the College of Science and Engineering, Scott Emmerich in the computer science and Mike Langston in computer science. So we have faculty not just in different departments, but in 3 different colleges. Right now we have a web page hosted out of the Center for Environmental Biotechnology that gives a list of all the courses we teach, just so that students have a resource to find the courses. But other than that, there's nothing really organized. I'm hoping to establish a computational biology intercollegiate minor next year. It's not something I can do this year, but something I'm looking at doing next year. It's also become part of the data science PhD program. One of their tiers is bioinformatics, but I don't know that much about the data science PhD program. So outside of that, I don't see it becoming a major anytime soon. I think a minor is probably the best we can hope for. Yes, Robert Murdoch runs that, and he's the one who keeps the website with all the courses updated, and he's a great point person to go talk to. And also similarly to Matt, he's available to help with projects. I think 50% of his time is devoted to consulting and helping students, faculty, and staff. So he's coming more from a microbial background, whereas I'm coming from more eukaryotic. So I feel like our skill sets are complementary and we enjoy working together. So yeah, definitely. So hopefully when you get out of this course, even though it's only 1 week and 1 credit hour, I'm hoping that you'll be able to apply some basic bioinformatic theory and tools to analyze transcriptome data sets. We are focused on a single type of data. But I think once you see one type of data, it makes it easier to kind of expand and apply those principles out to other types of bioinformatic problems. I want you to be able to communicate and critically assess the application of bioinformatic tools to transcriptome data. And the way we're going to work on this is by everyone's going to review a paper and try to think critically. And this is really important because even if you end up working with a colleague or a friend to do your bioinformatics, it's your data, you need to understand what's happening to it and if it's being processed correctly. And I'd like for you to have a basic competence in the Unix Linux shell and usage of bioinformatics tool from the command line. So hopefully that's what you will get out of this at the end of the course. I pointed you already to the course website. I will have office hours every day this week from 1:30 to 2:30 if you want to come by. And today, anybody in particular who's a little concerned about the amount of command line in this class, I'll be happy to sit down and go through it with you to show up at 1:30. And let me pop over to the syllabus real quick so we can go through that. It's linked right at the top. And of course, this only applies to the students who are enrolled. There's 3 of you who are not enrolled. So you wouldn't necessarily need to do any of the grading part. So I'll let you guys read most of this on your own. We already went through the learning outcomes and the objectives. The class will meet every day this week from 9 a.m. to noon. So it's not a traditional schedule, but we'll be in here every day. We will have a combination of me talking, other people talking, as well as guided computer labs where you're going to open your laptop, open a terminal, log in, and do the work yourself. And I think that that's really a powerful way to get you started with doing data analysis. And we'll also have some short group class discussions. If you at any point need help, we have tons of people here. We have 4 people from my lab at all times. So feel free to raise your hand. Even if we're in the middle of a lab and you're stuck and you can't move forward, go ahead and raise your hand and get help. And I don't mind if you guys are having like a side discussion over here while the rest of the class is moving forward on the lab, because sometimes if you get stuck and you get too far behind, it's gonna be really hard for you to catch up. So it's better to just go ahead and raise your hand and get help. Everyone, please arrive prepared. We will also attempt to arrive prepared and be respectful. I've never had any problems with that, but it's always worth saying. Classroom environment is a place hopefully where everybody comes and feels positive and is having a good learning experience. Outside of office hours, feel free to email me anytime. You can also email anybody in my lab. Office hours 1:30 to 2:30. My office is right down this hall on the right. It's 154. So basically, for those of you getting graded, it's a pretty simple point scale where the number of points you get indicates the grade. It's pretty straightforward. Attendance is going to get you, if you come every day, you get 25 points. So that's an easy just show up and you're already a quarter of the way there. We'll also have each day 1 output file from the lab that you need to email me just to show that you completed the lab and you were able to get through the work. And I've only made it through Tuesday as far as the course material being updated. So don't worry about looking too far ahead. But when you come in each morning, I will have the full day schedule and the homework posted. So just email me that output file, and that's going to get you another 5 points a day. So you're halfway to your total points. At the very end of the course, what we're going to do is a journal article review, and you're going to write up a summary and kind of give me, and there's a little bit more information below, but that's going to be up to 50 points to your grade. So that's really the primary thing I want you to focus on doing a good job at. And then if you take the course survey, so you will get an email from the university saying, please evaluate this course. If you do that, I will give you an extra 5 points. So there's actually 105 total available points. So the journal article writing assignment. So I have it right now, and we can stick with this. Each student will select a peer-reviewed journal article about RNA-seq analysis in your field. So pick something that's interesting to you. Pick something that's relevant to what you're doing. And write a 2-page summary and a critical review of the experimental design and analysis methods. I'm not necessarily really interested in the results in the discussion. What I want to see is that you're able to apply the principles from this class to understand the paper and to critically evaluate if they applied computational techniques appropriately to the data set. It's really fun to pick a paper where they didn't, and then you get to tear it apart. But I want to see both strengths and weaknesses of the paper. Is the experimental design robust? So this is kind of the rubric right here for you to look at. And then as far as getting points for the end of course survey, those answers are anonymous. I'm not going to be able to see who filled it out. So once you fill out the whole survey and you're done, it should say like, thank you for filling out the survey. Take a screenshot of that and email it to me. So I will never be able to know. You can completely trash the course and say it's terrible. I will never know it's you. But I will know that you filled out the survey, which is what's important to me. Because I really want to know, was this helpful? Was it too fast? Was it too slow? Was it good, bad, whatever? And everybody in the lab, we end up going through all of the comments and really trying to improve the course every year. So a lot of students, I have a really terrible rate of students filling out the survey most semesters. So this is a new thing I'm doing to try to get more responses. Because we really do care. We really do want to know what you think. Yeah, you get points for attendance, but if you don't attend, like I'm not really, that's up to you. You can still pass the class and not attend. I will post the videos every day. My recommendations for how to be successful include following along with the exercises during the class period, because if you end up trying to do them in the afternoon and you don't complete them and you come in the next morning and you're already behind, it becomes a huge problem. The class just moves really fast. So keep up with things during the class period. And if you start getting lost or getting stuck and something you're doing isn't working, go ahead and raise your hand. Get help early with problems. Come to office hours. If you're getting behind, let us know. And select a journal article that is a proper scope for the writing assignment. If you select a journal article that doesn't have any transcriptome work in it, that's going to be a problem for me to grade. So if you have any questions about whether the journal article you're thinking about is appropriate, let me know, or you can just send me the article and I'll let you know. And I think I have the due date, I think it's a week from Friday. And I think that's somewhere in here. Yeah, August 9th. So once the course ends, you'll have until August 9th to turn in your review, and then I'll grade them real quick that weekend and post grades. Yep, and the rest of this should look pretty familiar because that is the boilerplate for all of the syllabi at UT. But I always encourage students, visit with what resources are available to you and utilize them because they're there for a reason and they're all free. And there's a lot of really good stuff out there. So definitely think about using all of those. Okay. Back to this. Okay, so what are the stickies all about? The stickies are kind of a way for me to judge if people are having problems or if they are good. So if we do like an in-class exercise, I may say put up a green sticky when you're done, put up a red sticky if you're stuck. Or if we're going through a lab and you think that we need to slow down, just put the red sticky up on your laptop. And when I see a sea of red stickies, I will know that we need to slow down. Also, if you don't want to raise your hand, just put a red sticky on the edge of your laptop and one of the TAs in the back will come up and help you. So it's just a way for me to kind of judge if things are going poorly or if they're going well. And it's a way for you to like very discreetly say, hey, I'm stuck, I need some help, and somebody will come help you. And then at the end of the day, I'll get everyone to write one thing they liked and one thing they didn't like and turn the stickies back in. So I'm going to go ahead and start talking about the basics of RNA-seq, and hopefully Nick will get here in the next few minutes and then he can talk about the ACF. So this lecture might get broken up in two pieces a little bit. I want to go through the big picture. The data we're going to be using is Illumina. Why are we using Illumina platform? Why do people mostly use Illumina for RNA-seq? I'll give you a little bit about FASTA and FASTQ format. And I can't remember if I left the slide for the data set for the FAST, but maybe. If not, I'll just talk about that ad hoc. So the big picture, what are we trying to do here? Just as an example, this is a project that we're about to start on in my lab where we're looking at how berries develop. We're looking at how blueberries in particular go from kind of like hard, green, not very tasty berries to sugary, antioxidant rich, blue, soft berries. How does that process happen? And so the way we're going to look at that is transcriptomes. We want to look at gene expression. So in this case, and this may not be the case for every project, but in this case, the genome between these two berries is identical. It's the same plant. So the difference in between the two has to have something to do with the genes and how the genes are being expressed. So you can think about this as the green berry has, those cells have copies of the genome. The blueberries also have cells with the exact same copy of the genome, and the exact same genes are available for use. But if you do RNA sequencing, what you're going to get is the number of times those genes have been turned into transcripts, the number of times they have been transcribed. And so the question becomes, what's different between these two samples? So if we look at the red gene, we're going to say, okay, it looks like the red gene is getting much more highly expressed in green berries, and it's more lowly expressed in blueberries. That green gene, it looks like it is not expressed at all in the earlier stage, but it's starting to be expressed in the later stage. And then the final gene, the blue gene, is the expression different? I don't know. It's only slightly altered. So this is where statistics comes into play. How are we going to figure out what is statistically significant and what's not? So this is just a very, very high level overview of like RNA-seq. And this is another diagram that I think is really helpful for thinking about it. There is transcription and intron splicing going on in the cell in vivo. We can't see any of that. All we're getting is that mature mRNA. In vitro, you do a lot of molecular preparation, including converting to cDNA and turning that into sequencing. And then what we're really interested in this class is the in silico, which is you've got piles and piles of data. You now need to use a computer to figure out what that data means. If I said alignment versus assembly, how many people feel like they have a pretty good idea of like what aligning a read is versus what assembling a group of reads is? Everybody feel like, yeah, pretty good on that? Maybe. Okay. And we can talk more about that too. But that's kind of going to be, if you have a genome sequence, you're going to align your reads to it. If you don't, you're going to assemble your reads. We'll talk a little bit more about that tomorrow. Sure.
In terms of what I'm doing right now with the microbiology, it's kind of like the boundaries are kind of gray as to what the biology does and what the microbiologist does, and turn up there.
Yeah.
Near that bottom rectangle that says which. In other words, what's the roles and responsibilities in terms of what the bioinformatics or what the microbiologist handles? Is the bioinformatics ahead of the microbiologist?
I think it just depends on the lab. Like there are some labs that are extremely self-sufficient and they do everything from extracting the RNA all the way to their own analysis. There are some labs that are very collaborative where, you know, one lab does down to the sequencing and the next lab picks it up and does all the analysis. I think the goal moving forward is for emerging PhD students or master students to have the skills to do both. Because a lot of biologists who were educated in the 80s and 90s, we didn't really have big data in biology. And they didn't learn how to do this. And that's not great. That's not because they're dependent on other people to ask research questions to help them answer their research questions. My goal for you guys and for all students is that you'll be able to do everything. That you'll be able to do the field work, the lab work, and the computational work. This class is only going to be the in silico. I'm going to talk super briefly about how we do library preparation, but that's not really my expertise. We're a computational only lab, except for Fong. Fong's my only student who does lab work, but mostly we do just computational, and that's all the course is going to cover. Oh yeah. I mean, I've definitely had people tell me that it's not you're not a real biologist because you're a computer scientist, but that kind of attitude is changing because so much of biology is now pinned back to big data and being able to process your data. You're not going to get as many people who say that. Okay, I will, I guess, pause here and we can take a quick break and I'll get Nick set up on this computer. This is Nick, he's from the ACF. He's going to be talking to you about how wonderful our high performance computational systems are and how you guys can make maximum use of them. So let me, do you have a thumb drive?
I do not have one.
Could you email me the slides real quick? I'm recording, that's why.
Okay.
It's easier to just like keep it recording on here. I should have told you this last week and I forgot. I had Bobby's from last year and I realized you're probably not using the same ones.
I actually have the same one. Okay.
Well, if you do, yeah, if they haven't been changed from last year, I have the PowerPoint from last year. Does this look like it? Okay. Yeah, you can just email it to me. So while we wait for a minute, I'm just curious, do you guys already have transcriptome data that you're working on, ready, or just out of curiosity? Okay, what organism?
Also that table kind of
Okay. Who else? Okay. And you're in fact, yeah. And you'll have data. Yeah. I think some of this will apply. Yeah. You can get more comfortable with command line.
I'm working with Dr. Bruce on cellulose production.
Are you guys also on the same project? Okay. Cool.
Recording off of your laptop.
Yeah. Okay, let me start the screen share. All right.
Awesome. Hi, everybody. My name's Nick. So I work user assistance on the for the ACF. The ACF is the Advanced Computing Facility, and it's managed by the University of Tennessee. And it's housed at Oak Ridge National Lab. That's where all our staff and faculty reside for the ACF. So I'm going to go over the ACF today. Everybody here probably has accounts. I saw a lot of requests last week for everybody applying for the project for this class. So if you go here, if you haven't applied for a project yet, you just go to Getting Started. The website's nix.utk.edu. You just request an account. If you already have an account, you can use the same link to request access to a project, which many of you all have done already for the project for this class, which was 0085. I won't go over some of this stuff. So the ACF, it's available for everybody who has a UT Net ID. As long as it's active, you can use the ACF for as long as it's active for. And you also have the ACF or your Net ID active a year after you graduate, so you can still use it after you graduate also. So we have, we only have one way now to SSH into the system. We don't have RSA anymore, so that needs to be changed. But so if you go to, I'll show you real fast. Everybody is comfortable logging in or not. So if you're using a Mac, we'll do SSH. It's going to be your net ID at acf-login.nix.edu. And your net ID password. You'll be prompted with the duo push. Everybody's probably fairly comfortable doing that. Because everybody, I guess, attempted. You don't necessarily have to follow along with me if you don't want to. I'm just showing you how to log in. Yes, so I can help you log in if you have account issues. Your account might be disabled due to inactivity. That's the most common cause that your account might be disabled. You'll get like a little message that'll say currently unavailable, and then it'll kick you out after you SSH in, and that'll be the reason why. So if you have a Windows machine, you have to download a SSH client. PuTTY is the most common one. It's free. It's a very, very small download. It just takes like 30 seconds. I won't download it on here, but basically a box just like this will pop up. And the only difference is this box right here says host name. And instead of typing SSH in your username you're just going to type in acf-login. .next.utk.edu here, and you're going to keep it on SSH of port 22, which is the default. And then when you hit open, a terminal box is going to open up, and then it's going to ask for your username. Then you just type in your net ID. And then you'll be prompted with the Duo login, and you'll be in the system. So that's how you SSH into the system. So I encourage everybody to go to the website, next.utk.edu, and go to computing and go to ACF. And this is all of our documentation right here. It's all separated in these tabs. So you have your system overview. It just tells you all the nodes and partitions in the system, how many cores, how much memory they have, etc. Access login software tells you some of the available software, data transfer. I'll go over that here in a little bit. File systems, running jobs. So all that's available on the website. Feel free to go there, read it. Yes.
ACF is the software that's the gateway into SDF. What's the software actually doing? So it's doing log on verification.
So you're talking about when you SSH? Are you talking about when you SSH? ACF. No, ACF is a high performance computing system. Oh, the ACF is a high performance computing system.
Oh, so it's actually
NICS and JIX manage it. So NICS is a National Institute for Computational Science, and JIX is the Joint Institute, and we are housed at Oak Ridge National Lab. So we're a UT entity that's not here out on campus. We're at the lab, and we have a computing cluster out there that we manage, which is called the ACF. So this is our system overview for the ACF right here. It's a, yeah. So, yeah, so these are a bunch of Dells. So they're a bunch of Dells. Yeah. It's a computing environment. So it's, we have about, I think about 300 nodes that the ACF is compiled of. So then we have a condo system that the ACF is broken down into. So UT pays money in it for their own institutional condo, which everybody who has a net ID has access to. It's just a pool of nodes that everybody has access to. Nobody has any special like privileges on it or any priority or anything. And then we have investor condos, which it could be inside or outside investments can pay to purchase their own investor condo. And they, no. So yeah, this is all local to our machine. So these are all Dells. So some of these are just different brands. Like these are Dell Skylakes. We have Dell. So we have Intel K&Ls. We have a Monster Node, Sigma Rho Beacon. Some of these were part of the Newton project that UT had a couple years ago. So OIT had their own system. It was called Newton, and they didn't want to support it anymore. So they gave the entire machine to us. They were just a bunch of nodes, and we took all the nodes and added them into the ACF.
Do you ever, for example, there's a piece of software that I've used in past where you get a CPU time. So in other words, we have things where you get a CPU time up, horsepower as compared to
So you're running some jobs and you're potentially getting some issues, CPU issues, and you're wondering that if the okay, so yeah, so whenever you're running, depending what the issue is, if I mean, if you're, hogging, memory, or if you're hogging IO or anything, you'll get messages back from the system. If you're the wall time is a little different. So if you're using the opportunistic project, you can run up to 24 hours, and that's the max you can run. So if your job requires more than that, it'll die at 24 hours. If that's what you're asking. If Oh, no, it's our model is a little bit different than that. It's so we used to run off an allocation based model that did CPU time and all that stuff. So we don't do that anymore. The condo model means that whatever resources that are available to you, you can use them as much as you want, as hard as you can. So um, yeah, you're not going to be like charged or bottlenecked by your CPU usage or how much you're using or the time that you're using it or anything like that. So yeah, so every computing center's model is all a little different how it's managed. So yeah, the previous one that you used it'll be different than ours and different than somebody else's, and so no, that's a problem. You want to run batch jobs? Yes. Yes. I will go over that. So awesome. No problem. No problem. All right.
Slower version of how to log in, and then we'll see whoever's having problems with it, try to help us.
Is there a lot of people having trouble?
We've got one person disabled.
Okay.
I don't know.
Okay. It only take it.
She always having a hard time about being kicked off every week. It says she hasn't logged in for 90 days, so she's logging in every week. So we don't know.
We have a few users that have that issue. No matter so the rule is if you're disabled, if we enable your account, you're enabled for 5 days without activity, and then if you don't log in, you're disabled at that 5 days. Then once you log in after the 5 days, you're good for 90 days. That's the way it's supposed to work. But we have some users that use it every day, every other day, a few times a week that still get disabled every 7 days. We don't. It's a bug somewhere, so we haven't exactly got that. There's not that many users that have that problem, just a handful. But
She's one of them.
okay. You're fine. Inactivity will bump you off. Like if it maybe like a 30 minutes, just like if maybe if you put it to sleep and turn it back on, it'll kick you off because that's a broken pipe. That's the most common error that you'll get if you're inactive. It'll just say broken pipe. It'll kick you off. You just hit the up arrow and it'll just get you back on again. All right, so when you first log in here, you're in your home directory. Everybody's home directory looks very similar to this. It's going to be Nix, some letter, home, and then your username. Everybody's limited 10 gigs on your home directory space. So you can use the quota command to see where you're at. So I'm at 3.6 gigs out of 10 gigs. There's a 2 gig grace, so you have 12 gigs total. This is an NFS, a network file system, so this cannot be extended. This is not a file system that you want to use to necessarily run jobs from. We have a lesser file system that I'll go over for that. But here you can just store all your scripts or small files, things that you're working. You can store that all here. So this is the path that you want to go to run jobs. This is the Lester file system. It's a parallel file system. It's really fast. So all your jobs, all your batch jobs particularly, you want to run here. So there's no quota limit on your Lustre space, but it can be subject to purge. Every 30 days it can be. So if you have any files, anything that has not been touched in 30 days that's sitting in here, it can be purged. It may or may not, but if you come on here, so you want to back this up regularly. As soon as you get jobs and you get all your, your outputs and whatever you're doing, you want to take this stuff and back it up out of your scratch and just move it somewhere else. So you have your two main file systems. Let's see what else we got here.
If I do a CD to most of the people use the same mix of
So Lustre, so CD space / so Lustre Haven user and your username for the ACF.
That always trips me up. I think it should be small.
So you know it's going right if you use the tab button in auto tabs.
No, you should automatically get that user directory in an account, right?
Yes, you automatically have a Lustre directory. So her issue is she didn't put a space between the CD and the /
This is all like really every else has don't worry, we're gonna go through like super basics next.
No, you're fine.
If I want to share something from my user directory, you should also know
You wanna share files? Yeah.
what makes help share a sequence.
Okay. Yeah. No, that's fine. We can go ahead and do it. So I'm not in the project, so everybody that when you request a project and requested to be added into a project should have requested the project for this class 0085. So I believe that project does have a project directory, so everybody can see the project. Everybody that's in that project has access to it. So I think it's the path will be Lustre Haven Proj. Oh, is it courses? Yeah. Yeah. So I guess I do have access. Yeah. So everybody can CD into this directory, and this is a shared project directory. So you can put stuff in here, and somebody else can come and look at it or whatever. So this directory path is open for everybody in this class only. So no one on the outside can get to it. So yeah, no problem. So yeah, so the path, if you didn't catch that though, it is Lustre Haven courses and then the EPP.
We're not going to do a great job of covering the Linux command line in this course, but for those of you who, this is a permission thing, right? Like this is the whole thing. I can spend 3 hours on permission.
Spend longer than that.
And we're not going to do any of that in this course. Unfortunately, I can teach a whole semester-long course on all that command line if people do. But there's great online Coursera courses, or like we have lynda.com as part of the subscription. So I can point you to places a lot more familiar with Linux, how to navigate it. Plus, we're going to do a lot of copying and pasting on the command line. And some of it you'll just have to kind of explore how that works in your
So I want to introduce everybody to their portal page real fast. So if you go to open a web browser, you go to portal, portal.nix.utk.edu. You'll come up to this page right here. So you just log on, UT user, just your, just going to be your NetID and your NetID password. And again, you're going to be prompted with Duo. So this is going to show you your profile information for the ACF. So just a few things I'll go over. So projects down here. So everybody will be in the project 0011. And then everybody should also be in the 0085 project for this class. And if you're ever taking any additional other classes in the future and you have professors that also have their own projects, you can click on this link right here under projects and you can get their project ID from them and you can hit submit and then it'll come to me. And once I get confirmation from whoever owns the project, you'll be in that project. So that's how you can request to be added to another project. Besides that, if for some reason, like in your Q subline or in your batch script, if you don't want to specify your project, you can come here and you can change your default project. So whenever you submit, it'll read a project and it'll say, can't find a project, we're just going to use your default project. So you can come here and do that if you like. So you can set it for 0011, 0085. So you can come here and do that. Here's where I check if you're enabled or disabled. So you can also come here and check. It'll just say disabled right there, and you know that you'll be disabled. And then also, if you want to be subscribed to the mailing list, I send them out usually every Monday afternoon. Just hit that checkbox and hit submit. So it's an ACF mailing list. You're more than welcome to subscribe to it. Yes, default is bash, yes. Yes. So you can change your shell on here also. So we got Z shell, C shell, all these other shells. You can change it on there. If you do decide to change your shell and you get a bunch of errors or weird stuff, just send a help ticket. And I'll show that here in a little bit, how you can contact us. All right, file systems. I'll go over one method of data transfer. So everybody has access, should have access to globus.org. So in order to connect this to the ACF, you go to login. And UT is not going to be on this list right here. So you're going to sign in with the Google account. So you'll need a Gmail account. And then you'll just log in with your Gmail account. So this will probably be the easiest way for everybody to transfer your data, because this is a GUI based. It's pretty easy. So you're going to have, so this is a screen that's going to come up. So one thing that you'll need to download, you go over here, it's called Globus Connect Personal. So this is this right here is what creates an endpoint to your own personal machine that helps you connect to the ACF. So it's a free download. It's quick, and it'll pop up on your toolbars on your computer, and it'll say it's active on there. So you'll need this installed on there. All right, so for this right here, under File Manager Collection, these endpoints, there you can also install Globus right there. So you want to make 2 endpoints, one to the ACF, and it's going to be one to your own personal computer. So when you're going through the Globus Connect, it'll go through a endpoint creation where you just name your own machine, whatever. I just named it my first name. It's anything that you find is active when it's green right here. You know it's active, so you can click on it. It's gonna, so this right here is going to show me everything that's on my own personal computer. And then all right, so this is the panel page right here. So here we're going to hit, so the data movers, they're called nix hash data mover, and then just use 1. So this is what it's going to look like right here. So it's going to have my Globus Connect on this side, which is going to have everything on my personal computer, and then it's going to have everything over here that's in my home directory, which defaults my home directory in ACF. So this is all everything on the ACF that I have. You can change your path here. So you can put / Lester Haven user, your username. You can put Lester Haven courses EPP for this course. And all you literally do is click on something. So this is the ACF, and I want it to my personal computer. I click it, I hit start, and it just automatically transfers. So this is really nice. I can go over here, click this, and transfer it. Once this is done, so it'll tell you if it's succeeded or not. So this is a tool that you want to use for data transfer. There's others if you want to learn the command line, if you want to learn SCP, SFTP, and all that other stuff, you can. All right, so this will be the QSO command that you'll use for this class. She will have the exact one that you'll have. So basically, it's QSO-I for interactive, -A for your project. It's going to be ACF.UTK0085-L add res spring. Actually, you're going to take the spring res out, I believe. Yes. So she will have the command for you though.
And you click on that home tab, the very first tab, it's for all the way to the bottom of the shape that we're using if you want to use that very first tab in the browser window.
Oh, okay.
All the way to the bottom. There's the QSub command.
Yep. All right.
Yeah, it's kind of long.
It is long.
But we can copy and paste it. That's hard to put it.
All right, yeah, so this is the QSO command that you'll be using for this class. So this should work. It takes about 20 to 30 seconds to launch a job. So there is a 2-node reservation for this class for the debug queue. So with the N-access policy, everybody should be able to get on at the same time, hopefully.
So we'll talk a little bit more about this, but basically once you SSH into a login, mode, you're on one computer, but that computer is being shared by everybody on the media. You're not going to run jobs on that. You're then going to basically use QSub to request access to a different computer where you're allowed to run computational jobs. Come two-step process. Log in, request access to a computational node. And it's important, and we'll have it like the top of every lesson. Make sure you're on a computational node and not a login node. If you're on a login node, everybody does not like you because no one can log in. So we'll talk a lot more about that. But that's what's going on here.
Not sure why it's. It depends on the scheduler and like that the system is using. So we use Moab and Torque, which is a scheduler and job placement for our system. But other did get on. But other computing systems that other organizations might use something different. So like at ORNL, Titan also use Moab and Torque, the same scheduler and job placement that we have. But if you have like something like Summit over there, they're using IPM's own proprietary scheduler. So it'll be different. So QSub it'll be just for anybody, any computing group that is using Moab and Torque. They're not, commands will be different. So there's many job schedulers out there that have their own commands.
So you'll be able to access them. So, and we'll see some examples of this. ACF is like its own contained computation system. But then it's connected to the internet. The login nodes are connected to the internet. So you can say, grab this file from NCBI. We'll see wget. And you can suck in the data on the command line. But you won't be able to log in to NCBI. So it's more like if they have to have exposed the data over the internet.
All right. So like I said, you'll get the QSub command later, but so it's going to wait for your job to start. This is going to be your job ID. So every single individual job that you submit is going to have its own individual job ID. No matter how many you submit, it's going to be its own. So when you do an interactive job, you're going to land right on a compute node. So here we're on beacon 008. So you're already on it. You don't have to SSH into it. So here you can go and just do all your stuff. You can use the top command to see everybody that's, so if everybody use top that's on this node, everybody's gonna be able to see everybody on this node. So that's how you'll get on the system. Using the QSub command there. Yes. No, you're fine, you're fine. So yeah, like I said earlier on that, no, if you're doing it from if you submit from your project directory for this class, I don't believe purging is set. So purging should not be set on any project directories at all. It's only on your scratch base, which is your Lester Haven. So that's, like I said, that's only going to be purged potentially every 30 days if it's not touched. So. So here's if you need to send a help ticket, help@nix.utk.edu. That's where all of our help queue tickets come in. I receive all the help tickets. So if you have any questions and you need help to send a help ticket, you can also email my personal email. It's ncsercse@utk.edu. How do you get out of your terminal? Oh, hit Q. And then if you're in a terminal and you want to get rid of all of it, you can just type clear. So if you do top, you're in here, you'll be here. If you hit 1, it's going to show you everything that's active. 1 takes you back, and Q it gets you out. If you're in a node, you just type exit, and then you're out of the job. Is everybody fairly comfortable with editing? No, you're fine. You're fine.
The maintenance period where we probably should go in because
So we do have maintenance periods. So we do in the ACF announcements that's sent out every Monday. I put that in there. So our next outage isn't until I think the first week in October. So it's it's a while. Usually they usually range from 2 to 3 days, and then we bring the systems back into production again. So yeah, it can get about a month and a half for our next outage. Yeah. So we do have 2 different editors on here. We do have Nano. If you like to use Nano, I don't. I never use it. And then we also have VI. So here, I'll show you actually when we get into a file. All right, so this is something that's happened. So if you want to edit a text file, you just hit VI in the name of the file. If you haven't created a file, you can just hit VI and just space in the name of whatever you want to create. This is already a file I already had, so I wanted to show you. So when you first come into VI, you're in something called command mode. It'll let you scroll down, look at stuff, do all this fancy stuff, but you won't be able to do any editing in command mode. So you can type I, and that's going to bring you down to insert mode. Here's where you can do all your editing and changing and stuff. So I can go over here and delete stuff, change stuff, go down, do whatever. If I want to comment, you can comment stuff out, uncomment stuff out. So you can do all that in command mode. VI is very difficult to learn. It's very difficult. Yeah, he knows. So it's there's a lot of different. There's so many different commands that you can do to manipulate your files. You hit escape to get out of command mode again. You can also do stuff like DD deletes an entire row. If you're done with the file, you hit escape. You hit Nano. Okay, I won't go into this much, but you do command colon. I figured. If you just want to quit, you just hit quit. If you want to save it, you do right quit, and I'll save it. But yeah, she'll go over Nano with you then.
Yeah, it's great. If you're going to be using the computational, like you're using command line or your or yours, it's where you're.
Yeah, if you plan on using Linux.
But it's amazing.
Do you have any trouble with the module avail?
I haven't.
Okay. Okay. Yeah, there was a permission error. So, so if you want to know all the available modules, software installed in the system, you do module avail, and it's going to show you everything that's installed. So if you If you want to load it, you just do module load in the module name. And you can also just add that to your batch script also. Yes.
Is there a help option? Can I do
I believe so. Yep. And also, the man pages are very helpful. They'll be your best friend in Linux if you're not very comfortable. So man stands for manual. So you can do man and then space in any command name. So you can do man ls. It's going to go and tell you every possible way you can use the ls command. So every command has options. It's going to go through. So I would highly recommend using the man for any command that you might be stuck with. Man cd. It's gonna tell you everything about every command. So that's a very helpful something to know. I'll go over any of that. That's about everything that I was going to go over. Does anybody have any questions, or is there anything in particular you want me to go over real quick? Yes, you're fine.
Nick, is a smart way to do it to use an external editor like Python and then copy those into a nano file or a VI file and save it next to the syntax on what you use for the external.
Yeah, once you're comfortable with Nano, I just write down everything I'm doing in the terminal, and we can shape that. Or you can save it as an external file. Some people prefer to keep like basically a electronic lab notebook. For right now, but we do it in the terminal.
It's a very good thing as well as, like, say, you submit your results and the computer asks questions about what you did. You'll have that external file to go back and for reference.
So one of the things I was hoping you would touch on, Nick, is so a lot of times I get students who take a class and then all of a sudden they get like, their data arrives and they have like 500 gigs of data, and they really need to scale up to have more storage, maybe more priority. Can you talk a little bit about how that works and how to buy in?
Okay, so if you need more storage, the easiest way is to get your advisor to create to request an account in the ACF, because only faculty and staff can request projects and project directory spaces, any type of spaces on the ACF. So have your advisor, whoever, create a project and request a project directory. During the form, they can fill out how large they want the space to be. And once it's created, it's not an automatic process. It's created by one of our staff members. You just need to be added to that project, just like you were added to the project for this class, 0085. It'll be a different project ID. And then you can start storing all your stuff in there. Like I said, you can keep on storing all your stuff in Scratch. It's not one of those places where you want to permanently store your stuff, though. Then we don't increase home directory spaces because it's NFS. So that's limited to 10 gigs. So the best way is to have have an advisor request a project directory space and store all your stuff in there. We can also increase the project directory for this project also. It's not a problem. So I don't know what we have it set to right now.
I think it's right if we go with that, it should be
okay.
We
All right.
the data set, but we wanted it to run fast.
But that's for students, that's pretty much the only way is to get your advisor to request a project directory space in a project. We do charge, I think it's $42 a terabyte is for a year, and it's $42 for backup for also as per terabyte. So
That's a great price.
that is a good price.
And I think you can also buy computational time. So if you need your job to run priority, basically buy an hour, right?
Yeah, you can. So if you're faculty or staff that you're working for, so what they could do is they could buy their own condo. So and what that means is that you can buy a node, purchase multiple nodes. We have many faculty and staff out here that have their own private condos. So a typical node costs about 10K. A GPU node costs I think 18K. And these are brand new nodes. These are Dell Skylake. They're 20 core or they're 40 core, 198 gig nodes that you can buy. And if you do decide to get a GPU, they're the NVIDIA Volta GPUs. So they're nice $8,000 GPUs. So, but
So that you could segment your job to go to a GPU or just a regular.
yes, you can. So on the open side, we don't have. We have I believe 2 GPU nodes on the open institutional side. So if you want to, I'll show you real quick how you can. So if you go to running jobs down here, just go all the way down to the bottom, you're going to have all these partitions and features and stuff. So in your job script or your Q subline, you can do -L partition equals, and then I believe we have 2 Beacon GPU nodes that's in the institutional cluster. They're used a lot. So sometimes the wait times can be kind of long on them, but so we do have 2 of these available on the open side for everybody to use. So like I said, anytime, just send a help ticket if you have any questions, you need any help on anything, just send a help ticket. Yes.
In terms of code content, what do you not want to do? What's a stupid thing to do?
We, I mean, so we do give warnings if you run jobs on the login node. I have some tools that monitor login node usage. So we do have 2 login nodes now. So in their round robin, so they're called 5 and 6. So what I'll do is I'll do top on the login node, and I can come here and I can see if anybody is running on the login node. So I'll keep, like this person right here is running MATLAB on the login node. He shouldn't be, but so I'll usually email the user. You'll get a couple warnings, then you'll get a disabled account, and then I reach out to whoever the person is. That's probably the most common thing. We have a lot of users that try to run on the login node, which bogs the login node down. Other than that, users don't usually get in trouble. So we did have an incident a couple weeks ago, and it was public. We disabled the command to do it, but there's a wall command where you can publicly post a message that goes out to everybody on the system. And this person said some bad words, so we had to. They were mad because a user was running jobs on the login node, and they were really bashing the login node. So we ended up disabling both their accounts. We had to reach out and do UT code of conduct policy and all that stuff like that. But yeah, so yeah, we have disabled the wall command, so you can't go on there and post your mind. But so that happened a couple weeks ago over the weekend, and it went on for a while between several users going back and forth at it. So I've never seen anything like that before. It was pretty funny, but but yeah. I was sitting here doing all the screenshots for it, and I was like, oh gosh, I turned to my boss, and he turned into leadership, and it got messy after that. So
Just treat it like a shared resource. And if you're doing something wrong, we all do something wrong at some point. People like know.
yeah, if like any questions, any problems, issues, you just send a help ticket in. They all come to me. So that's the best thing. We'll go through and look at everything. But yeah, that's everything I have. Is there any additional questions that anybody has? Yes.
So I don't actually have access to the 0.085
Okay.
project. Okay. And don't know if we should like bombard you.
No, I can do it all here. It's no problem.
Nick, what I was thinking, if you have a few minutes to hang around, I can get everyone to, we can start over, get everyone to log in and queue sub, and everybody who can to, and CD to the course directory.
Okay.
And anybody who fails along the way, maybe you should try to help, and we'll run around.
Okay, that's fine. So if you're not in the project, just go to portal.nix.utk.edu and log in with your UT net ID and password and do a push, which takes you to this page. And then go down to projects, click on this. You want to request to be added to an existing project. So you'll click on that. The project ID for this class, ACF-UTK, if I can spell it right, 0085. And it's hit request access, and I'll have to get on my computer and just approve them. So anybody who's not in the class, go ahead, or in the project, go ahead and do that.
And I did fill out no ticket response.
Awesome. It was probably me then.
Yeah.
Yeah. I mean, all the staff and faculty see the tickets, but I'm responsible for them. So I usually answer about 95% of the tickets that come through, which is a lot some days. Yeah, so everybody just do that, hit request access. I'm already in it, so we might get something. Actually, it'll probably still pop up for me, but. All right. I can go through and start proving everybody real fast. You don't need to do it one time. Some people did it more than once on here. All right, looks like everybody just went through and did that. So yeah, I think give the system like maybe 15 minutes, 15 to 30 minutes, and you should be in the project. So when you're on your portal page, you'll know you're in it when it pops up in your projects. So when it pops up down here, you'll be in the project. So it might take about maybe 15 minutes. Shouldn't take too long, I don't think.
So let's take like a quick 5-minute break. And then we'll come back. I'll lecture for a few minutes, go through some more of those slides, and then we'll try to log back in if we still have a problem. I do know before you leave, Nick, we have people who are disabled.
Yes. I got it.
All right, be back at like 10:30.
Who is disabled? Come up here and see me.
Thank you.
Great job. Thank you very much.
No problem. No problem. All right. What's your what's your first name? What are these you right here? You should be good to log in now. Go ahead and try it now. You should be good.
Okay. If I was in this class, how would I go about getting that?
Send a help ticket.
I'm the one who gets picked up every week. So I wanna, what should I do?
There's nothing that you need to do on your side. What's your username again?
Day1.
Oh, yeah. Alright, so you are enabled.
Yeah. Yeah, I'm able to go.
Oh, okay. Oh, okay. So look here. Disable 73, disable 79. Enable you on 79, 715.
So it will take 10 minutes for them to be added into the project, the course project, right?
Yeah.
Okay.
I don't know what's going on. I'll have an admin look at your account. See if there's something wrong with it. Besides that, I don't. Yeah. So it looks like every 5 to 7 days you're getting kicked out. There's but I'll let the admin know and see if they can find something. Just keep on sending out tickets. Yeah, there's, I don't see anything, there's nothing I can do about it, so I'll take it to an admin. Yeah, you're enabled. At least right now you are, so should be good.
Is there anyone else who knows if they're disabled? They log into the user order, it's like portal. Alright, if you can stay on 15 more minutes of lecture, then we can go through, try to get everybody log in and see if it works better. Now everyone's in the project. All right, I know that was a short break, but we got a lot of material to get through. So I'm just gonna start back in. Hopefully it's 15, 20 minutes. We can try again with logging in and everybody's part of the project. But in the meantime, I wanna talk to you a little bit more about RNA-seq data and how that works. So we in this class are going to be using Illumina data. This is the logo for the company. And this is, I would say, by far the most common type of sequencing data you're gonna see right now. Illumina is a company, has $3.3 billion of revenue in 2018. So this is a big company. They're making a lot of money. The market share for sequencing, when I looked it up in 2016, it was estimated that 90% of DNA and RNA sequencing in the world was going through Illumina machines. Right now, it's 75%. And I think these are mostly just estimates. I don't know how easy it is to actually try that. So what you're seeing is that there are some competitors coming on board to Illumina, and I'll talk about this a little bit. But Illumina is still dominating. So why are they so popular? Why have they put essentially everyone else out of business? They have a very low price, which is always appealing to academics, right? We're all very poor, so we like low price. High throughput. So Illumina really offers just like the most number of DNA bases for the price. High base calling fidelity. They're very accurate reads that you get back. And they were the first ones to really nail down and do a good job with paired-in sequencing. And I'll talk about what that is. If you are an investor, this is the Illumina stock. It is a publicly traded company in the United States. So you can buy a piece of Illumina for $304 as of July 24th. You could own a little tiny piece of Illumina. And as the stock price goes up or down, you could make or lose money. So the interesting thing that I've been noticing is sort of over here, it has not been on its upward trajectory since let's see, the end of 2018. And what happened there is that Illumina announced the acquisition of PacBio. So those of you who are keeping track of the sequencing market, PacBio is a competitor. They're a long-read sequencing technology. It's a very different type of technology. And it is currently being heavily examined by regulators, and it may not actually go through. So there are a lot of investors who are worried that this is a bad acquisition for Illumina, and there are a lot of investors who are excited about it, and there's also regulators who are concerned about it. So it's been very interesting to watch. So sometimes I like to point out that you can think about these things not just from a science side, but it's also kind of interesting to think about this from the business perspective. And this is an extremely lucrative business to be in. So this is for me was a sea change in my life. I got my PhD in 2007, and it was right about then that we transitioned from Sanger sequencing, which in comparison to what we have today was very low throughput and very expensive. A company called Solexa came on the market and had a completely different way of doing DNA sequencing. And this is what was really, we call it next generation sequencing, because it was a huge change. It was super cheap and it was super high throughput. Illumina was already a company at that point. They sold other types of biotechnologies. They bought Solexa. They took it, they made that instrument widely available, ramped up production. It was very expensive, but it was so awesome that lots and lots of universities and companies were buying it. And you can see from this chart what has happened to the cost per raw megabase of DNA sequence. It just plummeted. We're currently at kind of a stable position right now. It's no longer going down as much as it was. And part of this, I think, is that we've maxed out the improvements that can be made to the Illumina technology. And part of it is that Illumina doesn't have any short read competitors. There's no advantage for them lowering the prices, right? Why would they lower the prices just to lower the prices? There's no competition to, so that's kind of where we are. Since 2015. It's been pretty stable. But yeah, this kind of changed everything in bioinformatics. All of the software that we're going to talk about in this class has been written since 2007. Like literally not a single piece of software that I used in my PhD is now in use. So it's a little crazy how fast this moves, and you really have to be aware that a lot of these algorithms are still being improved, still being developed. It's a good idea to keep your eye on the literature. What are people in your field doing? What are they using? And this is sequencing has become so cheap that informatics is actually now a bit more of the bottleneck in many labs. You can sequence so much data that really the bottleneck is now owning the computers and having the computational expertise to sequence that, to understand and analyze that data. And it's even gotten to the point where some people propose that it's so expensive to just store the data, it's easier to store the samples and resequence them. It's actually cheaper to resequence the samples than it is to pay for the storage and the backups to keep the data. That's how much data we can produce for very, very cheap. So I want to talk a little bit about how Illumina works. I'm on a few committees here, graduate student committees, and I will tell you, I'm going to ask you about this at some point. I'm going to expect you to understand this. I'm not going to go through it super in detail in this class because it's a super quick class, but It's important if you're generating data, even if you're sending it off to a company and getting the data back, you need to have a good idea in your mind of what's happening at the molecular level. How does this technology work? Because otherwise you can't troubleshoot if something has gone wrong, and you may be looking at data and it has major flaws in it and you won't understand that. So it's really good to try to think about and understand this. So first thing, when you extract total RNA from your tissue of interest, if you were to sequence that, you would get probably 70 to 90% ribosomal RNA. We don't actually really care about ribosomal RNA usually, because it's the same in every cell and it's not that interesting. What we care about usually is mRNA. Now, of course, some of you may be into small RNAs. That's a different subject. We can talk about that some other time. But for the purposes of this class, we're going to assume that you're interested in messenger RNA, protein coding RNAs. So there's two ways that you can select mRNA for your sequencing so that you aren't getting all ribosomal RNA. The first one, which is what happened to the data that we're going to use in this class, is a poly-A enrichment. So if you have a eukaryotic organism, once the RNA is transcribed and spliced, it has a poly-A tail added to the end, so A, like that. And so you can just use that tail and have a bead pull down and literally pull out, enrich your sample for just the transcripts that have A's at the end. I mean, that's going to really make sure that you're sequencing 90+% mRNA, and you're going to wash away all the tRNA and rRNA and all of that. If you are not working with a eukaryote, you will not have a poly-A tail to work with. And in this case, or if you, there can be other reasons you might want to do this, you can do ribo removal, which is essentially, it's a similar process, but you're, instead of enriching for something and selecting for it, you're going to select for the ribosomal RNA and get rid of it. So those are the two main types of libraries you'll see. Yep, and that's just pointing out in this whole, when you go from the genome to the primary transcript to the mature transcript, that's when the poly-A tail gets added. Super basic introduction to library prep. You have your poly-A, you're going to capture that and or do your ribo removal. The next thing you're gonna do is fragment your RNA and random primer it, which we will see in the data probably tomorrow morning. Random priming is never random, but you're gonna attempt to do some random hexamer priming. You're gonna use that random priming to create cDNA. And that's just complementary DNA to your RNA transcript. And this is good because DNA is more stable, easier to work with, easier to ligate. So that's an important step. You will get rid of the RNA, create a second strand, a cDNA, synthesize it from your original template. There's some 3' ends are adenylated and 5' ends are repaired. And basically this is just preparing you to ligate on adapters. So this is just adding adapters to either end. They're getting added to each and every fragment. And this is what facilitates sequencing and makes sequencing possible. And then from here, most kits are gonna take you through a PCR amplification step. Some kits are PCR free. Again, this can vary a lot by kit. TrueSeq and Nextera are probably the ones I see the most. There are other kits out there, depending on what sequencing company you use, or if you use a university, they may have their own, they may not use a kit, they may have their own system. So you're probably going to want to ask about it. And the reason why I say this is because when you get your data back, these little adapters that have been ligated on are often left. They are left floating around in your data. So let's say I'm sequencing blueberries. When I go to process that blueberry data, I just want blueberry DNA. I don't want Illumina adapters floating around in there. So it's important to know that different kits have different adapter sequences, and you need to find out what those adapter sequences are. But this is what the fragments look like when they go onto the machine. There is a universal adapter, or this is just for TrueSeq, the universal adapter. Your fragment of interest, I mentioned that Illumina is very short for read sequencing. So 150 bases, maybe up to 250 bases. You can do preps of 500 bases. We'll talk about that. And then another adapter, and there may be an index in there. We'll talk about indexes too. But this is kind of what it looks like when it comes off of the machine. Most of this should be gone, but that is not always the case. In my 3 credit hour fall class, I make everybody watch videos, but we don't have time, so I'm just going to give you the links. It's really fascinating to see how sequencing works. There's cameras and millions and billions of little tiny particles, and it's actually really fascinating and interesting. There's also a really good library prep video by Thermo Fisher. Especially even if you were sending your data out and you're a graduate student, it's really important to still understand this and understand how it works. So before I mention paired-end sequencing, and I want to visit that a little bit, we're not going to use paired-end sequencing as an example in this class, but a lot of people will get paired-end sequencing. And so basically, if you have single-end reads, you're just taking these little pieces of DNA and sequencing them. For paired-end reads, what you do is you select a slightly longer piece of DNA, and you sequence from both the forward direction and the reverse direction. So you get 2 sequences that are paired. That's why they're called paired ends. There's usually a gap in between them, but you know how they're ordered and oriented against each other. So you start with 1 piece of DNA. If you're talking about transcriptome sequencing, it's complementary DNA. And you have all of the bases coming from the 5', maybe 100 bases coming from the 5' end, and then 100 bases coming from the 3' end. And those will be called read 1 and read 2. And they'll have a unique number so that you know they go together and they originated from the same fragment. And this is just a way that Illumina was cleverly able to get around their small read limitations. This gives you a little bit more information about, so it's basically like getting a longer read, but you don't know what's in the middle. So down here you can see, you can do this for anywhere from 200 to 1,000 base pair fragments. You can actually, there's something called mate pair sequencing where you can do much longer distances, but that's another story. So why would you bother to do paired-end sequencing? It is more expensive. You use more reagents on the machine. The library preps a few more steps, but not too bad. But it's useful because you can overcome lack of length. And this is particularly useful for mapping accurately to repetitive regions. So as many of us know, many repeats in the genome. You need the same sequence of bases multiple times, sometimes very close together. So if we imagine we have these 2 repeats, if you gave me this blue read as a single read, I wouldn't know if it originated from the first repeat or the second repeat. However, if it has a pair that is originating in a unique region, that is going to anchor this fragment into the correct location on the genome. So I will now know if there's some polymorphism in the blue read, whether it originated from the first repeat or the second repeat. So getting the extra length just really allows you to place the read accurately in its genomic context. You can also identify insertion deletion mutants. So if there is a huge deletion, your reads will map very far apart, and you will know that they actually originated from a piece of DNA that was very close together. You'll know that there was probably a deletion in your genome. This is also, if you were assembling a transcriptome from scratch, this additional set of information is really going to help you to do a better job with the assembly. Real quick, I want to go through multiplexing. With RNA-seq, this is critical because you most often want to do many samples. And the way you sequence many samples is with multiplexing or indexing. Those are kind of used interchangeably when you're talking about library prep. So envision, back to our original example where we had a green berry and a ripe blueberry. We could extract the RNA separately. It's in 2 different tubes. In this tube, we're going to do a library prep, and we're going to put in a pink index. So this adapter will contain the sequence CGATGT in this example. For the other RNA sample, we will go through the same library prep, but this time we'll use an adapter with a different sequence. In this case, I think I put TGACCA. The point is that they're different. They both have adapters that are going to allow them to be sequenced. They have these little indexes, these little sets of nucleotides in the exact same position, but they're different. At that point, you mix them together, put them on your sequencing machine, and sequence them. You don't actually care at this point who's getting sequenced where on the slide. They're all these fragments are just mixed together. you get your data back. 99% of the time, your sequencing company is going to do this for you. They're going to do something called demultiplexing, which is where they go look for those little barcode sequences, and then they send you a file and say, these are all of the reads that came from your pink sample. These are all the reads that came from your green sample. So that's how you can take 40, 50, 60 libraries, all different RNAs, and sequence them on the same instrument at the same time in parallel. Again, for most people, this is pretty transparent. They don't really know what's going on. They just ship their stuff off to the company and data comes back. But it's important to know how this works. One thing Ryan's lab works on is sometimes these barcodes are missequenced. So data ends up in the wrong sample a small percentage of the time, or the data can't be assigned to any sample because there's an error in the barcode. So these things do happen, and it's worth knowing that that's what's going on. There are many, many different Illumina instruments to choose from. The HiSeq or the NovaSeq is the hot new thing that's like super, super, super high throughput. This is just a screenshot I took from the University of Minnesota sequencing facility. I like to use them for pricing out grants because their pricing is very transparent and their pricing pages are super easy to read. I have never actually ordered anything from them, so I have no idea if they're a good sequencing facility. I assume they are. They have lots of instruments. But it's a good example because they have so many different instruments. I thought this was a great thing to show you. So you have so many decisions. What read length do you need? These are all paired in. That's what the 2 by 150 means. That's a paired in. You get 2 reads. Each one is 150 bases. It tells you the number of reads you need, or the number of reads you can expect to get back. So these NovaSeqs, as you can see, give you a lot more reads than the older generation equipment. And the cost is more. So more reads, more money. But this shows you the cost per million reads. So this is actually kind of astonishingly low. You can get 1 million DNA or RNA-seq reads for $3.90. Now you can't actually buy things in units of $3.90. You have to buy them in units of $8,764, which is why sometimes you do actually want to use some of the older equipment. But I think that's just a great example of how cheap things have gotten, and also the huge array of choices you have. We do a lot of sequencing through companies because it has just gotten so cheap. Oh, and I should say this does not include library prep costs. Library prep is in RNA-seq what's normally going to kill you, because anywhere from $200 to $300 a sample, which adds up fast. GeneWiz and Novogene are currently both running specials right now. Well, they will do library prep and guarantee you at least 20 million reads for $189 a sample. This is the cheapest I have ever seen. So that's pretty exciting that prices are coming down there. There are lots of experimental goals for if you're doing RNA-seq, and I wanted to talk a little bit about those because we're not actually going to go through all of these in this class. The first one is just getting a catalog of genes. So if you're working on an organism, you have no data on the genome or the transcriptome, you need someplace to start. You may go in and start sequencing just to know what genes are here, what genes are in my organism. From there, you can start looking at gene expression levels between treatments or development time course or whatever you're interested in scientifically. So at that point, you're going from just cataloging what genes are there to saying what genes are expressed under what conditions. You'll need to do some statistics to get actual differential gene expression. The great thing about sequencing over microarrays, for those of you who are familiar with old microarray technology, is that you can actually do all of that for alleles and splice variants. So what alleles do I have in my organism, which is the different copies of each gene, and what splicing variants. You can have alternative splicing of most genes in eukaryotes. So do I have alternative splicing? Is that alternative splicing under some type of control that impacts my scientific question, time course, treatment, abiotic stress, biotic stress, whatever you're doing. Another thing you might be doing sequencing for is to annotate a reference genome. We do a good bit of this. If you do sequence a reference genome from scratch, the best way to annotate the genes is to sequence some RNA. You may be doing variant discovery, so looking for SNPs or SSRs that you want to do some genetics, some mapping or QTL studies or association studies. And another great thing about sequencing is you can actually get to post-transcriptional modifications. So how has that transcript been altered before it gets turned into a protein that you cannot see from DNA sequencing? This class is largely just going to focus on differential gene expression levels. My lab has a good bit of experience with every single other thing on this list. So if you're looking for pipelines or advice, just let me know, because yeah, we've done all of this before. But differential gene expression is sort of the bread and butter. That's what I see the most often. It's worth talking about the limits of RNA-seq a little bit. All technologies, all science has limitations, and knowing those is really important for good experimental design and for good data interpretation. The biggest problem, I think, with RNA-seq is that there's a lot of bias that can get introduced. When you sequence those transcripts, they may not be exactly representative of your sample. And places that bias can sneak in is during reverse transcription, during PCR is a big one, and during fragmentation steps. And what this ends up looking like when you get your data back is you have depleted reads at your 5' and 3' ends of your genes, which can make it difficult to identify the true start and end if you're doing novel genes. Also, you can get some depletion of short. If the gene itself is very short and the transcript is short, you can get underestimated expression levels of that. And one of the ways to combat some of these things is to try to do a PCR-free prep. So if you are not limited by quantity or quality of RNA, if you can generate a lot of really good RNA, you can try a PCR-free prep, and that can make your sequencing a lot more accurate. You can also see some GC bias and some length bias. So just things to be aware of. And some genes are just difficult to accurately quantify. They're too short. Maybe they overlap. If you've got a gene on the positive strand, the negative strand that actually overlap, it can be difficult to assign the read back to the correct gene. Or you have gene family members that are 95% similar in sequence. It can be very difficult to figure out, I'm looking at a piece of an RNA transcript, did it come from gene A or gene B? It can be difficult. And finally, another thing you have to really look out for is experimental design. And I have a whole PowerPoint on that tomorrow. But combating some of these limitations really involve really good experimental design. Briefly, I do want to mention that you have other options other than Illumina. PacBio and Oxford Nanopore are currently the two biggest. They are both long read technologies. You trade long length for less accuracy. So Illumina is up to 250 bases long, 99.9% accurate. PacBio and Oxford Nanopore are more like up to 50 kb long, but only 85 to 90% accurate. So that means every 7th, 8th base is going to be wrong. So completely different bioinformatic algorithms, completely different software sets. However, this can be, if you're interested in RNA, this can be really great for getting full-length transcripts. Instead of getting lots of little fragmented pieces of your transcripts, you can get from the beginning to the very end. You do not have to do fragmentation. A lot of these preps have no amplification, so a lot less bias. However, this is significantly more expensive. If you don't have a reference genome, I would say this is definitely worth, and you're doing your own transcriptome assembly, this is definitely worth considering. This is also really great for discovering and profiling alternative splice variants. Because you're getting that full-length transcript, you can see what parts of what exon were included in the transcript. So that was a lot of information really quick. Any questions? Yeah.
I was using a sequencing measure called the phosphor data.
Mm-hmm.
And when I was doing that, it gave me the results for the phosphor confidence value.
Mm-hmm.
Do any of these give you a confidence value? So 90% sure the same match, 70% sure.
Yep. Yep, all of the software will generally give you some kind of confidence value. And it'll also tell you like, oh, this read maps to multiple locations, or this read maps to one location, or this read maps to no locations in your genome, or not so much with assembly. That's a more complicated problem, but for read mapping, you will get confidence values. And you can just talk about percent identity. So my read matches with 95% identity to my reference genome, or It just depends on your organism. So I work on highly heterozygous trees. I'm not super surprised to see reads only mapping at 90-95% fidelity. And RNA, it's usually more, but if you went to something, that doesn't have as much heterozygosity, you would expect to see less than 1% percent identity differences in RNA-seq. So it just depends on your organism. Yeah. All right. Before we do file formats, let's all try to log in. And then anyone who can't log in, we can harass Nick a little bit. I'm sure he's excited. It'll prevent us from clogging up your help ticket. Okay, let me go back to our wiki. All the way down to Monday. Okay, so we're gonna go to log on to the ACF. And everything here is pretty well documented so that hopefully you can follow along and copy and paste in locations where it's super, super long. Try to make this really big. Okay, so if you previously logged in, you don't have to repeat this, but if you previously had problems, quit your terminal and start over, start fresh. If you type uname-a, you should see something that indicates like this says msatan1.rome.utk.edu. This makes me know I am on my laptop. This terminal is talking to my laptop. Next, we're going to try to make this terminal talk to the login node that lives at Oak Ridge. So you will need to put your own username. SSH your username at duo.acf.tennessee.edu. If this is the first time you've ever logged in, it might ask you a yes or no question, like, do you trust this? If you should answer yes, because we trust that duo.acf.tennessee.edu is what we think it is. At that point, use your UT email password to log in. And you'll get this duo push. I always type 1 and hit approve. Anyone who has an Apple Watch, you can install Duo on it and just hit approve on your watch. I kind of love it. That's super convenient. You don't have to keep up with your phone. Okay, red stickies need to start going up, anybody who this did not work for. At this point, I'm going to type uname-a, and it's going to tell me ACF login 5. So as Nick was saying earlier, there's 2 login nodes. One name is name 5, one is name 6. The key word to look for is login. If you ever type uname-a and it says login, you should not be running computational jobs. There's another step. You've got another step in your process before you can do anything. And for us, that step is going to be this giant qsub command. I do recommend cutting and pasting this because it's kind of a lot, and if you try to type it out, it can be difficult to get correct. If you're having trouble with Control-V, try Control-click, and then it should give you a paste option. And this does take 30 seconds to a minute to get you on the debug queue.
Yeah, just try to slam it. It took a little bit over a minute, so I'm not sure why.
So it's worth mentioning that this will not work after the class is over. This is not the command you should be using after the class is over. I'm going to recommend you visit the ACF documentation. If getting from there's a lot to learning how to use the ACF and writing scripts and QSubbing them. If you are not quite there yet and you need to process data, come talk to me. I have a separate Linux server that we can just run command line jobs on and get things done and without going through the ACF. It's worth learning the ACF, but I realize it's like a lot to conquer if you're just trying to get your data processed. So let's see if I can draw a quick picture of what's going on here. Not the greatest marker ever. Okay, so we have many laptops here in the room. Yeah, that's not a great marker either. And then we are all SSHing into this login node. And this is actually at Oak Ridge. So we're going through the internet. And then the login node connects to these other computers that have various names. So this is our SSH step. This is our Q sub step. So when you first open a terminal, you're just talking to your own computer. When you SSH, your terminal is now letting you talk to this computer. We're all talking to the same computer. Shared file system, same thing. So that's why we can see each other, like Nick was showing us talk. There, when you do Q sub, it's sticking you onto another computer. When you normally Q sub with the ACF, you're going to get your own node. For the purposes of this class, we're all going to be sharing a couple of nodes. So we'll all be hanging out on these let's see. It looks like I'm on beacon 10. So you may or may not be able to see what computer you're on from the command line. But if you type uname-a, it's going to say, hey, you're on a Linux computer, and the name of that computer is ACF-BCO10. And in ACF parlance, I think that means it's a beacon node, right?
What part?
PC.
Yes,
beacon.
yeah.
So there's different nodes with different number of CPUs or different RAM or whatever. So this just tells us the node that we're on. And then we're not going to worry about the rest of the stuff for the purposes of this class. Does this make sense? Any questions? Okay.
Is everybody at the point where you should see a node as a prompt? Yes.
Good. Okay, and now CD. So CD is the command for change directories. And that just means I want to go, I'm currently, well, let's see where we are. Print working directory, PWD. I'm currently in my home directory. Then I'm going to go to our course directory. So I'm going to CD to Leicester Haven Courses EPP. Now at this point, if anyone could not CD to that directory, red sticky or hand up. Okay, raise your hand if you are all good and you are in this directory and you feel excellent. Okay, we are mostly there. So I think other than Allison, and
Well, permissions issue. Some students can't access the.
I'm feeling like that's going to be a project.
It's going to be a project thing. They might not be.
They can access courses up until courses. But
That's going to be a project thing. So if you're getting a permission issue, raise your hand if you're getting a permission issue. Okay. They don't need to SSH with the, no, it's just the Q sub includes the ACF UTK 0085, right?
Yes.
Okay.
So if they get the prompt that they're in the node, then they're in the project. Let's see.
How did you want information?
I think kids needed to get in
Into the folder, but you can't do it. I'm sure. I can log in.
Okay.
It's showing that you're. It might not be.
For those of you who have made it to this point, if you can make a directory with your own user ID. So I'm going to put mstate1. You should not put mstate1. You should put your user ID and hit enter. And then do an ls. And hopefully you see your name, your user ID somewhere in there. So mkdir is make a directory, and ls is list the directories. And then if that works, cd mstate1. So cd to your directory, not to mine. And now if you type print working directory, you should see Lester Haven courses, EPP 531 2019 summer, and your user ID. If this causes you any problems, and it's not related to logging in, and Nick is not already helping you, put up a red sticky. I bet that's why. Does anyone else in their command line prompt does it say I have no username? Are you having permission problems? Got it. Okay. I don't know why it would say I have no username.
I think maybe some of the accounts just haven't logged in. It's weird because he can see these in the project directory, which he should be able to do. He doesn't use sit on slide. So it might not. It could be a bug. You know, like
software just maybe hasn't updated the system yet. I don't know about him because I didn't see me too directly.
All right, well, I have a little bit more on file formats that we can talk about while we continue to sort out a few lingering login issues. Don't worry, once we get logged in, everything will get much smoother. At least I hope so. I probably shouldn't promise that. So a lot of dealing with your data is just having things in standard formats. And the beauty of open source software is that lots of people all over the world can write software to work on your data, and then you can try 2 or 3 or 4 different software packages. But for that to work, we all have to be using the same file formats. Like the reason why you can make a docx in Microsoft Word and send it to me and I can open it is because my computer knows what a docx is supposed to look like. It's the same thing in bioinformatics. You really need to have these file formats. They have to be standardized and they have to work. Otherwise, things won't work together and you can't share data with your collaborators. So there's a whole plethora of software formats that we're going to talk about in this class. Some more complicated than others. But today we're going to start with FASTA format and FASTQ format. These are the 2 most common formats that you will get sequence data. FASTA format is a much older format. It's kind of one of the original sequence formats, and it's pretty simple. So if you look at a file, it's going to look usually something like this. The key thing to pay attention to are these greater than symbols that appear on some lines. That greater than symbol indicates I'm about to tell you about a sequence. It's a header. It's the very first line of a sequence. It is always going to start with a greater than, and immediately after that greater than, there should be some kind of ID. If you're working with NCBI, the ID will often look like something kind of crazy with a bunch of pipes or lines in it, GI pipe 3156, whatever, and it'll have the accession number. If you're working with a protein data set or maybe something from your own lab, the sequence ID could be something much more simple, just a series of characters and numbers. The sequence ID, as far as the standardized FASTA format, it can be anything as long as it is unique to the file. So there should not be 2 FN640832 sequences in the same file. That would violate the FASTA constraint. So each sequence has a unique ID. Below that header line, so even though this is all kind of squished into a slide, you have to think of this as a single line. There's a new line, and after that, there is the sequence data. This can be multiple lines. It can have, or it can be a single line. It can be amino acids or it can be nucleotides. According to the specification, your file should either be all nucleotide sequences or all protein sequences. You should not have a FASTA file with a mixture of nucleotide and protein sequences. Yep. So, and also there should not be any empty lines between your sequence entries. Any questions about FASTA format? Yep.
So I've got a Python on the power program. What I'm going to do is look at that first line, put something in the end, put that way, start taking the
Yeah, so the first thing in a FASTA file, it should always be a greater than symbol. The very first character in a FASTA file, always a greater than. So that's the first thing you look for. If you were going to blast the sequence or otherwise look for sequence similarity, yeah, you would copy and paste what's below that header line to paste into wherever you need the sequence. Mm-hmm. Nope. FASTA format tolerates your sequence lines can be any length and they can have as many character returns as you want. The definition, then you know that you've arrived at a new sequence when the next line begins with a greater than. So that's how it works. This is a good bit simpler than this. This is FASTQ format. This is what you will, when a sequencing facility, or if you're using your own sequencing instrument, this is what you're traditionally going to get off of the sequencing instrument. It's a little bit more complicated to look at, but to break it down, every sequence is in a 4-block set. So every 4 lines is a new sequence. And that block, that 4-line block, will always begin with an @ symbol. So when you're looking through, you can look for these @ symbols as kind of the delimiter that's helping you to figure out where a sequence starts. And let's go through, I'm going to take one of these blocks and just run through it real quick. So imagine this is the first 4 lines of your FASTQ file. You have an @ symbol. You then have a sequence identifier. Again, this should be something unique to the file. It's usually a set of random alphanumeric characters. After that, you get kind of another optional description. In the case of Illumina sequencing, this will actually tell you what Illumina instrument in the world this sequence came off of. Most of the time, we don't end up using that, but there is sometimes important information embedded in there, although it's not required by the format. The next line is the sequence. It is nothing but the sequence, and it must be on one line. In this case, there can be no character turns. The sequence cannot be broken up over multiple lines. The next line is incredibly useless. This is why I think this is like an incredibly stupid, dumb file format. It must start with a plus sign. It may or may not have anything after the plus sign. Why is there a plus sign? Why is there a third line? No one knows. I have searched the internet. The internet does not appear to know. This line is useless. It is always there. It is always useless. Sometimes it's empty, sometimes it's not, but it has to be there and it has to begin with a plus sign. Don't ask me why. The next line are, yep. So base pairs would imply nucleotides. So the length, you mean this where it says length equals 41? Well, it just depends on whether, I've never really seen a FASTQ formatted file encoding amino acids. They generally only encode nucleotides. But to me, nucleotides and base pairs are interchangeable. Like a single nucleotide is a single base pair. Right, so amino acids are encoded by 3 nucleotides, but nucleotides and base pairs, I would consider that identical. So 41 base pairs or 41 nucleotides, either one could describe this line. Yeah. Okay, so we talked about our totally useless third line that's just eating up all your precious storage that you're paying Nick $42 a year for. That's okay. I'm only a little bit bitter about this line, but it will be there, and it will not be a valid FASTQ file without it. This final line is the one that just looks like total gibberish. So let's talk about that line. That is the quality scores. Scores are encoded as a single character to save space. That means it's a little bit complicated to deduce what they are. If you see a exclamation point, that means that the quality score for that base is a 0. So let's go back. We actually see the first character we see as a quality is capital B. So let's look for capital B. So that's probably like, let's call that 31. You can number these from 0 to 41. So let's call that a 41. So if I go back, this C character, the C nucleotide, has a quality score of 41. This A nucleotide has a quality score of, let's say, 40. This G nucleotide has a quality score of 42, or whatever, 30, 31, 30, 32. Anyway, you have to look them up. You have to look each character up to get the numeric quality score. If you see capital letters, that's a good sign that your sequencing is high quality. If you're seeing symbols like hashtags and $signs, that's your sequencing is very low quality. This is a mathematical formula. So an I is 99.99% likely to be correct. And a star, which is way down there, is only 90% likely to be correct. So it's basically giving you, the instrument is trying to give you an idea of how good a job it did figuring out that nucleotide. Like, I'm telling you that this is a C, and I'm really confident in it, or I'm telling you this is a C, I'm not really sure if it's right, if it's actually a C. Maybe it's a G, maybe it's a T, I don't know. So basically, the quality score is just giving you a confidence. There is a whole Wikipedia article that does a really nice job talking about this, and also talking about the fact that up until 7 years ago, there were like 4 different ways of encoding quality values, and it was driving everyone insane. But since then, we have settled on this single way to encode quality. So all your new sequencing data should use this exact way to encode quality scores. Any questions about FASTQ? Let me back up. So comment line, unique identifier starting with @, sequence, useless line with a plus sign, quality scores. Any questions about that? Yep. Usually, well, it's if you can upload raw data to NCBI and it's looking for this. But if you're downloading data from NCBI, well, it can be FASTQ or FASTA, just depending on what your FASTQ is almost always used solely for raw data. Maybe trimmed, usually raw. If you're downloading a set of genes or a whole genome or a set of proteins, they're always going to be FASTA. Yes. Yep. They'll both be. This is very much international standard now. Thank goodness. there were some years, 20 years ago in the Wild Wild West when there were like competing international standards, but this is actually like quite well agreed upon across the whole world now. Any other questions? Super exciting stuff, I know. Okay, I'm going to talk briefly about the data set that we're going to use, just so you kind of have an idea of the provenance of where it came from. This is data generated as part of a grant that my lab has. This is GLE's PhD project. So the grant is looking at dormancy in apricot and peach trees. So we have trees, it's really interesting. You can buy a peach tree that is adapted to live as far north as New York. And it will know to wait through many, many, many hours of cold in the winter before it wakes up in the spring. Or you can buy a peach tree that is adapted to live in Georgia, and it knows to wait a very short amount of time before waking up in the spring and blooming. So we call that a high chill variety or a low chill variety. And we're not actually, we have a few clues. We don't really understand how this works genetically. How do these trees know? How does that adaptation to the correct winter length happen? What evolutionarily is going on? And so what we have is data. We're going to use just a single tree, but basically we took floral buds and sampled them at multiple time points. So in this data set, we're going to be focusing on a 400-hour of chill time point and an 800-hour of chill time point. These are stages called endodormancy and ecodormancy. So the 400 is ecodormancy and the 800 is endodormancy. So the bud has transitioned from being not competent to flower, waiting for winter to end, to competent to flower, waiting for warmth. So we're very interested in this stage, and we're very interested in transcriptionally what's going on. So that's in a nutshell the data that we'll be using. We have subsetted it to be a very small set of the original data, and it will only map, it only reads the map to chromosome 1 of peach. Again, this is just an effort to make sure when we run commands as a class, they take 5 minutes instead of 45 minutes, because no one wants to wait all day for their commands to run. And in a real case scenario, these things do take a lot longer. But it should be a good data set to practice with. Any questions about that? It's more like somewhere in the middle, but tending toward higher chill. There has been a constant selection over centuries for a lower chill stone fruits. So this is true. So prunus, the prunus genus has apricot, peach, cherry, plums, all the stone fruits essentially. And almonds are actually kind of exactly like peaches. They look just like little peaches. So all of those have, they're all from kind of Eastern Europe, Kazakhstan, China. And so a lot of them have really giant native ranges already. So you can go out in the wild and find trees adapted to lower or higher chill as well. Yeah, that's kind of interesting. Okay, so who still is not able to log in and create their user directory? Everybody else is good? All right, well, we have got about half an hour, so I'm going to move into the BLAST lesson. It looks like the Linux review, we'll just do it at 1:30 today if you guys can come. If you can't come and you're really concerned about not having a Linux background, let me know. Let me try to sort it out. How many people have like used the command line before? You're thinking I can make this happen. Okay, so that's most people. We've got a few that haven't. How many of you think that you will come today at 1:30 to see command line? Okay. So if you don't know command line, don't panic. Just try to follow along, and it's kind of like this class, and I've read that this is a really effective way of learning, but some students respond to it better than others. We're just going to try to like drop you off a cliff and say, here you go, start typing on the command line, good luck. And then as you get used to it, some of the principles will start to click. But at first it's probably going to seem really frustrating and like you have no idea what's going on, but don't panic. It's something that I think if you just like practice and keep doing it and keep seeing it, it'll make more sense. And like I said, I can also point you to lots of Coursera courses that can help. I think we're down to only one student who's having problems. That's Ken. So depending on your schedule, you're welcome to leave or whatever.
So I
don't want to keep you here all day.
You still can't use that too.
Well, it's even working in the
Oh no. So I'm going to go ahead and move forward with the BLAST lesson. For the two of you, just follow along as you can, as far as like just watching, and then I can help you catch up later. Or, you know, I'm not going to hold it against you if you can't turn in the homework because you weren't able to do the lab, but we'll see. Hopefully this works for everyone else. So everyone go to the home page, find the Monday material, and click on BLAST Lab. You're going to want to follow along on the screen. Now, all of us asked for a 1 hour login time, which is the maximum we can get on the debug queue. So at some point after we've exhausted an hour, it'll probably kick you off the system. If that happens, just let me know. We'll take a pause. Everybody can log out, log back in. It'll just take a few minutes. Okay, so the beginning of every lesson, we are going to do PWD. Are you in Lester Haven courses, EPP 531, 2019 SU, your user ID? If you do not see all parts of this, when you type PWD, put a red sticky on your computer and get help. Otherwise you can be, and this is really essential because if you're in my home directory, if you're in my directory, you can overwrite my files. Or if you guys are in each other's home directories, you can overwrite files. Or if you're in the main course directory, you're just gonna litter it with all these files that other people are gonna have to look at. This is the one disadvantage, the way we have the course set up. It's kind of like we're all sharing a laptop. We're all using the same file system. So we have to be polite and try not to step on each other's toes. The other thing I want you to type is uname-a and just double, triple check. That should not say login. It should say Linux and something other than login. Probably acf-bc/a number. Does it need to be bigger? Are you guys having trouble seeing? I saw some squinting. Is that okay? Better? Okay. So again, if you do not see Lester Haven courses, EPP 531, your user ID, when you type PWD, or if you do not see something other than login node, when you type uname-a, red sticky. Okay, so BLAST is one of the most common bioinformatics tools out there. When it was published in the 90s, it was the #1 most cited paper in all of the 1990s. Like, I just think that's amazing. I've actually met one of the guys who's on the publication, and his like citation ranking is unbelievable because his paper gets cited all of the time and everything. And it was just basically a really fast way to compare two sequences to each other. It's algorithmically much faster than the previous methods we had. So it's great. You can probably, many of you have probably gone to NCBI and blasted like one sequence against a genome or one sequence against a database. But what I want to talk about is how the ACF and high performance computational machines in particular let you scale up. So you can blast tens of thousands of sequences against millions of sequences, and you can do it in a really speedy, efficient manner. So we're going to learn how to use command line blast instead of GUI blast. So GUI, for those of you who don't know, it's graphical user interface. So A lot of us who work on computers, we say GUI when we mean like, oh, it has like a pretty website where you can copy and paste things, versus command line, which is where you're just typing text on the command line. So I'm going to make a directory to hold my BLAST lesson, just so it doesn't clutter up all my files. So mkdir is make directory, and then cd is change into that directory. This is a nifty tip. I'm going to do an ls. In my directory, I have only one folder. So if I say cd, I'm going to type B and hit tab. And the computer's smart enough to know, this is called tab complete. The computer is smart enough to know, oh, well, there's only one thing you could be cding into. This is the only folder you have. So if I hit B tab, it says, yeah, you probably mean BLAST example. When you're typing pads, this is going to save you a lot of time. Okay, so now if we do PWD, we see that Lester Haven courses, EPP 531, M31, BLAST example is now on the end. So you can think of this as like you're on your desktop, you created a folder called BLAST example, and then you open that folder, and now you're looking inside that folder. We are going to download some data. Can we download some Beacon nodes? Will that work?
Which one?
Can you do wget from Beacon nodes?
Yes.
Okay, good. This is another one of those things where you might want to copy and paste, just because it's going to be really hard to get these long FTP addresses. And paste it, and it's working. So that was kind of quick. I'll scroll back up. So I pasted this command in. wget stands for webget. I think it just means go get a file from the internet. I could get it from my web browser, but I also can get it from the command line. In this case, I'm getting an NCBI file that they have made available online. It is a few cow proteins. It is not all the cow proteins, but it is some of them. wget then gives me a bunch of messages. It says, oh, okay, hey, I'm looking up where this NCBI place is. Cool, I found it, I'm connecting to it. Cool, I'm logging in as anonymous. You're just getting all these messages. The command is just telling you kind of how things are going. And then it says, okay, I'm logged in. And then if it's a really big command, it'll kind of be like 1%, 2%, 3%, like just like it is when you download your laptop. In this case, it was pretty fast. So it got to 100% in 1.8 seconds. So it didn't take very long. And it says saved. So that's your sign that it worked. It says saved, and it tells you how much data is saved. And now if you do an ls, you should see this file. And I'm going to copy and paste the other wget command. Carefully not get the rest of the lesson. And I'm going to paste that and do the same thing. And I get the save, and we can see the cells a little bit bigger. Now if we do ls, you should see both files. Sure.
Yesterday, I was on the grayed out area, and some of the grayed out area is
Yeah, sometimes a period means this directory in Linux parlance. Let me see if I can find one. Sometimes, yeah. There are often commands that end with a period. I don't think there's any in this lesson, but we'll get to ones where a period is totally what we want to do. Okay, let me figure out what we're doing here. Okay. All right, green sticky if you have files and you can see them. Yay. Yay, green stickies. Cool. Okay, so the next command we're going to learn. Let's do ls/lh. So ls just gives you a list of the files. LH gives you much more information. That's the L part. And the H part says make the size human readable. So if I do that, there's a bunch of permissions. We are not going to learn about this in this class, but as you become Linux experts, that I hope you all will become one day, this will become very important. It's going to tell me I own these files, so you should see your own username. can tell you what group. So we should all have TOG 2221, and that means that we can all see each other's files, share files, overwrite files. Dangerous situation we're in, but it's okay. We have 12 megs for this file and 5.1 megabytes for this file, and it tells us when we downloaded them. If the files are changed, that timestamp will be altered. So right now they have a .gz on your laptop. If you wanted to uncompress these, you would double click on it. We can't double click because it's a terminal, so we're going to use a command called a gunzip or gunzip. Again, I'm just going to type CO and hit tab. So there's only one file in this directory that starts with CO, so when I hit tab, the computer's going to say, yep, I know exactly what you're trying to do here. And I'll hit enter. Now if I do an ls, you can see that it has been decompressed. And I'm going to do that for the same thing for the human protein file. I'm going to do gunzip hu tab and hit enter. And now we should have both of our files uncompressed. Yep. Yeah, so this is why I like to teach like what do these files look like, because despite the fact that they're highly standardized formats, everyone tends to use a different extension. NCBI likes to use .FAA for amino acid files and .FNA for nucleotide files. They're both FASTA files, but they just use .FNA and .FAA instead of .FASTA. It's kind of irritating. And sometimes you'll see just .FA. So there's 4 possible endings that I know of: .FA, .FASTA, .FNA, and .FAA. They're all the same file format. Yes, it's actually, if you move it over to your laptop, you can open it in something like TextEdit. Yep. So while we're here, before we load the software, another good command to run in relation to Ken's question is head. So the head command basically just says, just show me what the top of this file looks like. By default, it's going to give you about 10 lines. I'm taking head, CO tab. So I'm asking for the first 10 lines of the cow file. Like, just show me what's in there. I have some wrapping issues, but if I scroll up, I should see that there's my greater than symbol, my unique ID, some other crazy information from NCBI that we may or may not care about, but it is telling us it's an E3 ubiquitin protein ligase from Bos Taurus, so it is actual cow data, so that's good. And then in this case, this is amino acid file, so we're seeing all our amino acids after that. So if you were to do a head on this file and say, is it, I don't know what .FAA is, you could look, you could do a head, you could be like, oh, that's a FASTA file. It's, yeah, the accession. So if you copy this and go paste it into NCBI, which we can do, it's gonna, oh, not the New York Times, NCBI. Thanks, Nick. I appreciate all your help.
So does anybody else have any issues besides Ken?
So if
Aaron.
we, couple of minutes, it still cannot queues up. It's waiting to start, but it never starts.
You were in though. Yeah. All right.
Right, so I'm just gonna search for this on NCBI and it's gonna find E3 ubiquitin protein ligase from Bos Taurus. So this is a real NCBI file. And yeah, so that's why these unique identifiers are really nifty. They can get you to the same information anywhere you are. Oh, I didn't mean to close that. I can go back to here. Okay, so the next thing we're gonna do, Nick mentioned that there's a lot of software already installed on the ACF, which is fantastic for our purposes. We can type module avail, which is what modules are available. If you hit enter, you're gonna get just a whole pile of different interesting stuff. And what's great about the ACF is that it's not just serving biologists. There's physicists, there's math people, there's chemists. So a lot of the software, I don't even know what it is. which is fine. Probably don't need it. But if I'm looking for a software package and I want to know if it's installed, I'm going to try to just search for the name of it. So I'll do module avail blast. In this case, it'll tell me, yep, I know about blast. The version I have installed is 2.6.0. Let's say I'm happy with that, and I'm going to say, while you're down here, module load blast. So you have to remember, just because it's available doesn't mean you have access to it yet. You have to type module load, and that'll say, hey, go get that blast program and make it available to me. It doesn't actually give you any feedback. It just says, okay, you know, if you don't get an error, sometimes no news is good news, right? If you don't get an error, you can make the assumption that it worked. So there's a bunch of different blast programs. I put this in here somewhere. Here we go. And it depends on what you're trying to do. So if you have a nucleotide query sequence and you want to compare it to a set of nucleotide sequences, which we'll call a database, nucleotide is BLAST-N. You have a nucleotide query and you want to compare to a protein database, that's BLAST-X. And protein to protein is BLAST-P. There's also two versions where it includes a 6-frame translation for you. Or actually, it's with this too. So to compare a nucleotide to a protein database, you have to translate the nucleotides into proteins, into amino acids. And the problem with that is you don't know which 3 codons are actually, like, which frame is encoding the protein. So these 3 are much slower than the other ones because it has to translate that nucleotide sequence into all 6 possible frames. And then it blasts all of those 6 resulting protein sequences against the protein database. So just a fair warning that that's a lot slower. Everyone's good with codons and frames and yes. Yep. So you just have to know, like, what is my query and what is my database? Nucleotide to protein, protein to protein. And then you pick whichever one you want, BLAST-N, T-BLAST-X, BLAST-X. I have to look these up every time. There's a pattern there, but yeah, I just Googled them. But what we're going to do is protein to protein. So we already did the head. We already did the unzipping. So this is just a little command line that you can memorize. This is just a command to ask how many sequences. So grep is a way to look for patterns in a file. And I'm going to design a pattern that says any line that starts with a greater than. Give me, oh, and I forgot the -C. So -C says count the number of lines that start with a greater than. So this is a whole language called regular expressions. Some of you are signed up for the Python course next week. We're going to try to get to regular expressions in the Python course. I love regular expressions. I think they're super powerful, so I'm very excited about that. We won't get to them very much in this class. I just have this memorized as a way to check for the number of sequences in a FASTA file, because when you work with FASTA files, you end up doing this like 10 times a day. So this is going to tell me that my cal.1.protein.faa file has 63,676 sequences in it. And I'm figuring that out by just saying, please count the number of lines that begin with a greater than. We know in a FASTA file, the greater than indicates a new sequence. So if we count the number of those header lines, we've counted the number of sequences. How do you guys feel about that? Good? Not so good? Sort of makes sense. So we can do the same thing with human. So grep means look for the pattern. As you find that pattern, count. That's what the -C means. The pattern you were looking for are lines that start with a greater than. The caret in this case is just a symbol that means lines that start with. And then I'm going to type HU tab complete. And we see that the human protein file is significantly smaller. It is 19,056 sequences.
All right, so everybody is good. And I'll email you later. I'm going to figure out this afternoon. If it all of a sudden just works, just email me, let me know. But if you still get permission to guide, I don't see any permission issues. I'm not sure what's going on, but I'll look into it this afternoon. No problem. Anybody else just have any questions? Send it up, take it in, and I'll get to it. All right.
Thanks, Nick.
No problem.
So there's a bunch of more explanation on this page if you want to go through and read a bit more about grep. For our super short example, I don't actually want to blast 36,000 sequences against 19,000 sequences because we'd be sitting here waiting a really long time. So we're going to get a smaller subset of the sequences. So I can give the head command a little bit more information by saying -n20. I'm going to tell it instead of getting the first 10 lines, which is what it gets by default, I want it to get the first 20 lines. Now if I type head -n20 and then put the name of the cow protein file, this is just going to spit all of the 20 lines onto the command line. So I'm going to hit enter and you can see it's just spitting lines onto the command line. What I actually want to do is put this into a new file. So upon looking at this, I could tell you that the first 2 sequences are on the first 16 lines of the file. You'll just have to trust me. So if I do head -n16, and then this greater than symbol is going to act as a redirect. This is saying instead of putting it onto the screen, I want you to put it into a file. And now I'm going to name that file cow.small.faa. In this case, it does not spit anything to the command line because it's putting it all into that cow.small.faa. If I do an ls, I should now have a new file called cow.small.faa. And it contains the first 16 lines that are in cow.1.protein.faa. So I've got 2 sequences in there. One nifty command to see the entire contents of a file. If you ran this on the cow.1.protein or the human.1.protein, it's going to spit the entire file to your screen. So don't do that. But the cow.small.faa is small enough that we can do cow.small.faa. So this will spit out the entire contents of that file onto our command line. So we can see, if we scroll up, that we have one sequence with a bunch of amino acids, and we have a second sequence. So this one was 3503, and this one is 1537. So we've got 2 different sequences in our file. Okay, so one of the ways that BLAST is super fast is it does an indexing. There's a pre-step to prepare your database. And for those of us who grew up having textbooks with like glossaries and indexes in the back, if you were interested in a term, you could flip to the back of the book and it would say, oh, you're looking for information on cats. Cats are mentioned on page 16, 36, and 112. So it's an index. It allows you to go find where in this much larger set of information are these specific instances of what you're looking for. So BLAST is something really similar. I'm gonna talk a bit more about this tomorrow, but just as like super quick, BLAST is basically taking your entire database and creating a giant index so that it can look up where things are really fast. But that means before we can BLAST, we have to make that index. And the command for that is make BLASTdb. And it needs to know some information. So the first thing we have to tell it is what is the name of our database? What is the name of the file that contains our database? We're going to use the human.1.protein.faa as our database. And then it wants to know the database type. In this case, it's a protein. So you could put, if you had a nucleotide database, you put NUCL. If you have a protein database, you put PROT. In this case, it's protein. Make BLASTdb is going to give us some information. For those of you, for those of you still writing the command, here's the command. It says, okay, I'm building a new database, gives me the current time, adding sequences from FASTA, added sequences. So this lets you know that it's completed because it tells you it added all the sequences and how long it took. So if you had the entire human genome or the Arabidopsis genome, this might take a few minutes, so you would be waiting. But in this case, this is super, in the scheme of things, this is actually a very small database, so it's pretty snappy. But now if we do an ls, we see a whole bunch of new files. So we have our human file, but we also have a .phr, a .pin, and a .psq. These are those index files I mentioned, and I'm not going to get into how they're formatted. for our purposes, it really doesn't matter. BLAST just needs to be able to find these files. So once you create them, just leave them alone. BLAST will find them. Okay, next I'm going to actually run the BLAST. So BLAST takes a lot of different, just like make BLAST DB took a lot of, needed a lot more information. BLAST P is also going to need some information. First, what is our query file? So what sequences are we interested in querying? I'm going to do cow.small.faa. So this is our two little sequences. Our database is the human. And for the out, I'm going to name, you can name your out file whatever you want. I have my own personal pattern that I always use for BLAST output files. I'm going to say cow versus human, small, BLAST results.txt. So it's kind of long. And a lot of people get in a bad habit, which is naming files like a.txt or blast.txt. And then in 3 months, when you come back and are trying to figure out what the heck is in blast.txt, you have no idea. So what I like to do is put a whole bunch of information into the file name so that when I come back in 3 months, I'm like, okay, I was taking the cow sequences, I was blasting them against the human sequences, and this is the file that holds the results. And I see I have a typo. Okay, so we're going to let that run for a few minutes. So this was actually pretty fast. We had 2 sequences. We were comparing it to 19,000 sequences, and it ran in like a second. If we do an ls, now we have our results file. How many people you have a results file and things are going well? Pretty good. Yeah, most people keeping up. Cool. Okay, so we can now go look at this results file, and I'm going to use cat. Now in this case, I only had to type up to cow underscore, because once I get to the underscore, the command line, there's no other files that have to start with cow.underscore. And so once I hit tab, starting from there, it'll finish it out. Okay, so that's going to spit all of that to the command line. And you can scroll back up, and it's lots of information. And this is a perfectly fine way to look at a file, but let's learn a slightly better way to look at a file. Let's do nano. N-A-N-O, it's called nano. And then once you hit enter, this basically creates a nice little text editor inside your terminal. So now you can use the down arrow and scroll through. And it gives you some more information down here. So once you're ready to exit this program, you type Control X. In this case, the caret symbol means control. If you want to save edits, you can do Control O. If you want to page up or page down, Control Y or Control V. So it's just giving you some information on how to interact with this program. But I'm just going to use the down arrow and go down. So it says, okay, I have BLAST P 2.6.0. So the first thing the output file tells you is what version of BLAST was run. And it gives you the reference. This is the most cited paper of the 1990s. So they would like you to continue citing it. Another reference, it tells you the database. And then it starts with the first query. And it gives you a list of all of the sequences from all of the human sequences that gave significant results. So our cow sequence, our first cow sequence, matches an E3 ubiquitin protein ligase from human. This is not surprising. We started with an E3 ubiquitin protein ligase from cow. I said, what is the closest gene in human? It is also an E3 ubiquitin protein ligase. Yay, evolution works. This is a good sign. Our BLAST is working. But there are some other matches. So there are multiple different proteins that have from human that have some level of significance. These scores along the right are giving you an idea of like how good that significance is. So we can see that top hit is a whole lot better. It's much higher scoring than all of the others. And if you continue to scroll down with your down arrow, there's lots and lots of hits with decreasing levels of significance. And at some point we'll run out of hits. I will show you next how to filter this so you don't get a bunch of insignificant hits. But this is just the simplest version. Okay, so we finally got, so after it gives you a list of all of the hits, it gives you a little bit more information about each hit. So it says, taking this best hit, it gives you the score. The expect value is a statistical score of how good the match is. A 0 is essentially perfect. It is definitely biologically related. You keep scrolling down, it'll give you the actual amino acid to amino acid alignment. So the query is that cow sequence, and the subject is the human sequence that it's reporting the match for. So you can see for the most part, these amino acids are almost identical. There's a few changes. If it has a plus sign, that means that these two amino acids have similar physiochemical properties, even though they're different. So again, just giving you this alignment. As you scroll down, you'll get to the next match. Oh, and there's a region with a lot less similarity. So definitely more differences happening here. Okay, so that was the first cow sequence matching to the best human. And then it goes on to the next one. And so for every single significant hit to the human genome, or to the human genes that are in our database, it's going to give you that information and the alignment. So you can see this is our second best, and it is much less good. There is definitely a lot less sequence identity going on here. Does that make sense? Okay. So to get out of this and to get back to the command line, I'm going to hit Control X. And it pops me right back to the command line. So if you're trying to run BLAST and you want to figure out how to kind of alter your BLAST search or make it, customize it, you can type BLAST p-help. And it's going to give you a whole bunch of information. Like BLAST is a very, very flexible program. And there are many, 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 many ways to alter how it works. I recommend for any piece of software you use, you should go read about it. If there's paper, read the paper. If there's help pages, read the help pages. If there's a manual, read the manual. Because most pieces of software have a lot of options, and knowing how and when to use them is really critical. In this case, we're not going to use most of these, and I have spent time reading this, so I will just tell you the ones that I think we should use. The first one is e-value, and this allows me to filter for only very, very significant hits. I don't want things that are like, oh, 20% identity. I want things that are only extremely significant. So I can set an e-value. And let's play around with the out format. Because the output format I just showed you is extremely verbose. It was huge. It takes forever to scroll through. Sometimes I want something in a much shorter, more compressed. Oh, it's 12:01. Okay, so we will pick back up with this tomorrow. For the homework, instead of sending me BLAST output, send me a screenshot of your terminal, type PWV and take a screenshot and just show me that you've managed to log on to the ACF, Q sub in, and get to our project directory. So take a screenshot of that and email it to me, and you're all good on the homework. Today, 1:30 Linux review. We'll knock that out for those of you who need it. If you don't worry about it. And then I will see the rest of you in the morning tomorrow.